Hello, welcome to the practical in the plant life course, in which we are going to assess how plants undergo the most important transition during their life, during which sometimes do once, during which they go from vegetative to reproductive, and how this is controlled by external inputs. In our case, we will assess the most important external input, which is light. Light directly, or light as a measure of the time of year. What is the biological problem? The biological question, if you want, is a plant is developing through embryonic, if you want, stem cell-like cells, meristematic cells, throughout their life, and is producing new organs, new leaves, through what we call a shoot apical meristem. So that shoot apical meristem will, at its flanks, be producing a leaf, then a leaf, then a new leaf, then a new leaf. At some point, this plant undergoes a transition in which, instead of the shoot apical meristem producing leaves, it produces floral meristems. And as a result, the plant begins to produce not new leaves, but new flowers. Plants like rosette plants, like model organism Arabidopsis, at that time, they don't only change in that instead of producing leaves, they produce flowers. They also produce a shoot, because the leaves try to hold the ground, prevent other leaves, other plants from taking their ground. But once they flower, they want to eventually disperse the seeds, so they also produce a shoot. So this is a major transition, life stage transition, which for plants like Arabidopsis can be carried out only once. For other plants, for trees, it can be carried out many times, once a year, but it has to be the right time of year. Therefore, this is controlled by environmental inputs. And the most important environmental input is, in fact, light. Light as a way of assessing the local environment, but also, as we will see, light as a key element to, together with endogenous time, help decide what is the time of year. Are we in spring? Are we in summer? Are we in autumn? So the practical is going to look at how these environmental inputs determine flowering time. And we are going to do so by looking at the phenotype of two different plants which have mutations, loss of function, if in individual light sensors, photoreceptors. The plant that we will be using, as we did in the first lab, is Arabidopsis thaliana. You know the plant. It forms a little rosette of leaves that if you want, hold their patch. And at some point, the time is right for these plants to undergo the transition from vegetative to flowering. The shoot meristem, which was producing leaves, I can see is beginning to form little tiny flower buds. In a few of them, a little bit of shoot elongation, stem elongation has begun, and some flower buds are opening as real flowers. A few of these plants have begun to flower. Most of them seem to me have flower buds, are undergone the transition, have just undergone the transition. This is a wild-type plant. These plants, Arabidopsis, have a genome, the first sequenced plant genome, of about 28,000 genes. As it turns out, it's a very similar number of genes to the human genome. 28,000 genes, more or less. Here are two strains of Arabidopsis in which one of those genes, one in each, has become faulty. In these plants, one gene has acquired a mutation that makes the protein that would be synthesized unusable. If I recall, there are the early stop codons, so only a fragment of the protein is produced. So one gene has become mutated here. Still, the other 27,999, if you want, genes are well done. Yeah. A single gene is mutant. In this plant, another single gene, not the same one, a different single gene is mutant. Again, the other 27,999, including the one that was faulty here, are normal. The gene that was faulty here is normal in that plant. So, two genes are faulty. What do those genes encode? They encode two different photoreceptors. Although they are both light sensors, we can already see that the phenotype is different. These plants, while normal plants have just begun to flower, these plants, there is no trace of flowering there. The meristems are producing leaves still. They are delayed, they seem delayed. They haven't undergone, they have no clue that it is time to flower. While these plants seem to be a bit spindly, a little bit paler. Actually, they are made of few leaves. They are not such bulky rosettes as the others. And many of them, looks like most of them, in fact all of them as far as I can see, are flowering. And actually, they are a little bit taller. That probably means that they have been flowering for longer than the wild types were. These seem to have flowered earlier. So here we have, these plants are missing a photoreceptor, if you want, are semi-blind and are flowering early. While these plants are missing another photoreceptor, are also semi-blind in a different way, but they seem to be flowering late. So we tell you what the photoreceptors that we are looking into are. They are one kind of phytochrome and one kind of cryptochrome. And in the lab, what you have to do is understand, tell us, what is the function of those photoreceptors through your analysis of the mutants. On the one hand, you will quantify the flowering time of the mutants. On the other hand, you will carry out a molecular assay which will determine which one is the phytochrome mutant, which one is the cryptochrome mutant. How will you carry out the quantitation of the flowering time? Well, the simplest, most accurate way to do this is as follows. Because depending on the conditions, from temperature, changes, mutations that slow down growth, etc., plants may be developing faster or slower without actually the transition from vegetative to floral being the primary defect. Flowering plant biologists noticed that the most reliable way to quantify consistently flowering time was to quantify the number of leaves that the pseudapical meristem has produced before it undergoes the transition. So you will quantify the number of leaves that these plants possess at flowering, and that will be a matter of taking plants and counting how many individual leaves had been produced in this case before the flowering occurred. We are talking about the large leaves because the tiny leaves are actually these sorts of leaves of new shoots which are emerging. So the large ones are the first ones, 1, 2, 3, 4, 5, 6, 7 in this case. You will do this for a number of these plants, for a number of wild type, and you will obtain similar data for one mutant and the other mutant. We will explain to you how you will do this virtually through pictures that we will provide to you, and you can collect a few pieces of data, each of you, but when the whole class puts those data together, they provide a sample which is large enough to say reliably, look, this is the wild type, this plant clearly flowers earlier than the wild type, this plant clearly flowers later than the wild type. So you have a flowering time phenotype. How can you tell which photoreceptor mutation has caused those phenotypes? For that, you will do a molecular assay. And that molecular assay, we could call it a genotyping assay. We are looking for the genotype of those plants. How do we do this? We have those plants. You will actually obtain from a small amount of tissue these leaves are made of cells which are packed full of DNA. So you are going to do a DNA prep from these plants. The wild type, mutant A, mutant B. Let's imagine this is the wild type. These are cells of the wild type. These are cells of one of the mutants. What do we want to know? We want to know whether this mutant carries a mutation in the phytochrome gene or the cryptochrome gene. How can we do that? The genome of the plant is huge, 28,000 genes, 140 million bases. Our gene is very small, is 3,000–4,000 bases, and only one of those bases has changed. How can we look at that? Easy. We can extract DNA. You will carry out the extraction. You will be shown how to carry out the extraction based on affinity purification. It will be fun. You have DNA from your wild type and from your mutant and from your other mutant. Then you target a gene that you are trying to understand whether it does or doesn't have a change, to amplify it by PCR. What is PCR? You know, it's a means of copying a small section of the genome thousands, millions of times, generate multiple copies of this. How do we generate multiple copies of exactly the gene that we want? Through primers, which start the replication by the tag polymerase. So we will provide primers that target our gene of interest, let's say phytochrome. They target a region of the phytochrome gene in which in the mutant there is a mutation. In the wild type there isn't. With these primers we amplify DNA, this region of DNA from the wild type. With the same primers, but with a different template, a different DNA template, we target the same region in the mutant. We have millions of copies of that target sequence. Now, that target sequence has one single base change in the mutant, not relative to the wild type. Now, we have identified a pair of scissors, molecular pair of scissors, a restriction enzyme, which can cut the wild type sequence, but not the mutant sequence. So we take the PCR products and we subject them to enzyme digestion, restriction enzyme digestion. The wild type PCR products will digest a little, not a huge amount. The mutant PCR products will not digest. We will have those digested or undigested products in a tube. How do we know that the digestion has or has not happened? Obviously, gel electrophoresis. We run those two products side by side on a gel, undergoing gel electrophoresis. Because these fragments have been cut and have become of a shorter size, they will run faster in the gel and they will run further away from where we deposited them, the well. On the other hand, the mutant, which has the mutant PCR products, which have not been digested, will not run as fast, and will not run as far. We will, when they have run enough, we will take a picture of the gel using a fluorescent dye that binds the DNA. We will take a picture, we will analyze the picture. Aha, it is the right size that we were expecting, but this one is is larger than that one. This was the mutant. And then we compare, this was the mutant, this was wild type, what was the flowering time of the mutant, and we can deduce the function of the gene. If, for example, the phytochrome mutant were to be the one which is flowering late, we would say phytochrome action is necessary to accelerate flowering. If it was the cryptochrome gene that was flowering late, cryptochrome action is necessary to accelerate flowering. And with a little bit of further information that you have in the lab book that we will discuss in the course, we will be able to assess why different photoreceptors are needed to assess different features of the light environment, to interpret them as different clues. For example, competition, for example, the right or the wrong time of year. Thank you.
So yes, now we are going to collect sample for DNA extraction. So we have 3 genotypes, wild type, A and B. So first we'll collect sample for wild type. Basically 2 or 3 green leaves should be enough. I think 2 is fine because they are bigger. And now we'll flash freeze them in liquid nitrogen. It is very cool and difficult to handle with just one pair of gloves, so I'm wearing few layers. So we'll take out the sample with this tool and a small beaker. So now what we are going to do is crushing the tissue with this plastic vessel. We crush them in fine powder. Now we'll add the buffered AP1. So this is the lysis buffer. It will break the cells and allow the DNA to solubilize into the buffer. And it also has the RNase enzyme because we want DNA, so it will also degrade the RNA. Now further mixing. You shouldn't see the big leaf chunks. The solution should become the green homogeneous. And then stop it. So I see green solution here. Now quickly vortex. Now I'll do the same step for other 2 samples, but by changing the tips, it will cross-contaminate the sample. The last sample. is also done. And the next step, we will incubate this at 65 degree to lyse the cell for around 10 minutes. And we will meanwhile will come and invert the tube to mix thoroughly. So now the incubation period is over. We hope that cell lysis happened. Now the second step is precipitating the protein polysaccharides which is inside the tubes. So to precipitate, we are using AB2 buffer, 130 microliter. So we'll straightforward add this into each tube. And then mix. Because I'm adding from a distance, so I'm using the same tip again and again. I will put the sample on ice for 5 minutes in order to precipitate. So the incubation period is over. Now we will centrifuge this at 13,000 RPM and we'll get a clear pellet for the further step. It's crucial to balance this area. So in the next step, we'll collect the clear lysate into the square shaded purple columns. Now this will filter the cell debris and we'll collect the clear lysate which will be collected in this 2 ml collection tube. Debris are here. Without disturbing that, we collect the clear lysate. So I have already labeled the tube, as you can see. So we take 400 microliter of clear lysate from the respective tubes, for example this one. Without disturbing the pellet. Then I change the tip. Centrifuge this again for 2 minutes, maximum speed. So yes, the soap the debris are filtered through the clear shuttle column. Sometimes we see a small pellet, so we need to avoid that. And this step will collect the clear lysate. So I have already labeled 1.5 ml collection tube here. So we'll remove the column from here and see where is the debris, because you don't need to take that. So that was from the well type. Now, we'll add the preconditioning buffer in order to give the right environment to the DNA. The amount is 600 microliter. That's the buffer. You see some DNA threads. I'm not sure if it's visible from there. Now we'll take 650 microliter of this solution and then put this into this affinity columns. Now this has the power to bind the DNA onto the membrane. centrifuge this again at 8000 rpm for 1 minute. Very closed. So I have already set this to 8000 rpm. Wait for 1 minute. So we'll take out the filters and do this step again with the remaining solution. So there's a flow through that we'll discard because our DNA is on the membrane. Putting the remaining solution back into their respective tubes. Simply putting this back again. So let's take out the sample. We're hoping that DNA is on the DNA making columns. Now, time for washing the column. So we have the washing buffer. So according to the manufacturer protocol, we have to add 500 microliter of washing buffer. Each column. And then we centrifuge this for 1 more minute at 8000 rpm. So we did washing twice. In the final step of washing, instead of using 8000 rpm, we increase the rotation, which is 13,000 rpm, in order to completely dry the membrane. Now this is the elution step, where we will elute our DNA. So I have already labeled 1.5 ml tubes. Now we have the elution buffer. according to the protocol, it says to add 50 microliter dilution buffer. So yeah, first thing first, remove the columns and put them in 1.5 number tube. Yes. So now I put the dilution buffer at the center of the membrane. Now we'll incubate at room temperature for 5 minutes, and then we'll centrifuge at 8000 rpm for 1 minute, and then our DNA will be eluted into the 1.5 mm tube. So yes, we waited for 5 minutes. Now it's time to centrifuge and elute our precious DNA. So same rotation 8000 for 1 minute. And then once we get the DNA, we'll prepare it for the PCR reaction. So for that, we had lyophilized beads which contains the tagged DNA polymerase. This enzyme works at high temperature, for example 94 degree, and then this has nitrogenous base and the suitable buffer which is suitable for the DNA to amplify. Now we have used water 20 microliter to each bead, and because we have two assays, one for Cry2 and one for 5B, phytochrome B, so we have aligned the samples that way. Now in the next step, we'll add the DNA 2 microliter that we will obtain soon, and the primer mix. So we have forward and reverse primer for each genotype, which is Cry2 and phytochrome B. We got the DNA. So I'm just discarding the filters because we don't need it. Now we can keep the DNA sample on ice. So now we need 2 microliter of the primer mix. So I'm doing for assay 1, which is Cry1. Putting primer mix into each tube. So because now everything is same, so I'm not changing the tip. Similarly, we do for the second genotype in the set, which is cytochrome D. Two methyl in each tube. The last segment, adding the DNA. Split the vertex quickly. So I'm adding 2 microliter of DNA. And then we'll mix later. We are done with one set. Similarly, we will do for the other set. Make sure to change the tip. Now we'll mix the make sure they are settled at the bottom. If not, then tap. So we'll set the PCR machine. I think the program is already set. So which has 3 steps. One is denaturation step, or when the 2 strands of DNA, because it works at 94-degree temperature, so they pull apart. So that's denaturation. And the next step is annealing, where the primer binds to the DNA sequence, and the third is amplification. So this will repeat for 40 times. We have set the program for 40 cycles, which will be done approximately in 2 hours, and then we'll do the next step, which is restriction enzyme digestion, and then electrophoresis to check the assay. I'm just simply putting the tubes inside it. Make sure they are closed well. So now our PCR product is ready. So we'll take it out. So we are going to detect the mutation. So first we will do restriction enzyme digestion, and that is followed by gel electrophoresis, which will visualize the DNA bands. So basically, what is restriction enzyme digestion? So these enzymes, they have a specific recognition site and they chop off the DNA from there. So basically they are like molecular scissors. They have certain temperature for its activation, and also they require buffer to create a right environment for its functioning. So there are different types of restriction enzymes. Basically we select based on their recognition site, matching with our template DNA. So we have here two restriction enzymes for 2SE. One is CRY1 and other one is phyto-B. So basically we'll take 15 microliter of restriction enzyme. It has the buffer also, as you can see, green color, and then we'll mix with 10 microliter of template. Quick mixing so that at the end we can mix with the enzyme and the buffer. For the first enzyme, first assay, which is for CRY1, it requires 55-degree temperature, and then this enzyme gets activated. And the second assay, it needs 37. So now we'll give around 1 hour time for its proper functioning, and then we'll do gel electrophoresis. So now we are going to run our sample on gel. So this is done. The gel is already set. There's a comb. We'll remove this gently by holding the sides of the gel tray. And then in this electrophoretic unit, we'll pour the buffer. Gel. The gel should submerge inside the buffer, so I'm pouring it again. So the orientation of wells should be towards the minus, as we know DNA is negatively charged, and this moves towards a positive charge. Now first we load the ladder, which has a known size of DNA. This will work like standard. So 5 microliter of ladder is sufficient. Quite small amount. So you can turn this. Now this is the tricky part, the loading, because there are small wells and you should be very careful. So basically hold your hand with the other hand and go inside the well, and then you will see if you move backward or forward, you will see that you are in a case, and then release it and then take it out. That's it. Now we'll run the samples. It's good to have the way that you're going to load your samples. For example, in this booklet, there's already the orientation where we are going to load our samples. So I'm going to follow that. First is A1. So I'll align that accordingly. Alright. time you need to change the tips. So you need 50 microliter of sample to load. I'll mix quickly, 1 to 2 times. That's it. So I have loaded samples for assay 1, and because there are 4 extra wells, so I'll give space before I load for the next assay. So this is done, and I can see buffer has submerged the gel, so I'll close it now. So I set, so there's an on button, I'm pressing it there, and then we should see some bubbles. Yeah, I think I can see it. So now I'll leave it for around 15 minutes to allow the migration of DNA. And as we know, the bigger DNA molecule, the bigger chunk will take longer time, just like elephant compared to deer, to move faster. So 30 minutes is, I think, enough time, and then we'll see the DNA bands. This gel contains the a dye which binds to the DNA. It intercalates with this, and then it creates fluorescence under microscope. So we'll see DNA bands. So yeah, we'll wait for half an hour. So it's been like 30 minutes. We have given enough time. So we'll stop the gel running. And as you can see, the blue tracking dye has moved towards the bottom. That's the right stage to stop the run. Now I'll take the gel out of this device, get the extra buffer out, and then we'll see this under the UV light. So now is the time to see whether our product is digested or not. So what we are expecting here for wild type, I'm talking about assay 1 first. So the size of wild type is 335 base pair, and because the restriction enzyme recognizes the sequence of the wild type, it will digest to 309 and 26 base pair. The second chunk is very small and it's hard to see that, so we will only visualize the first 309 size of the DNA. And for Cry2, because there's a mutation, so the restriction enzyme would recognize that site, so it won't digest the sequence. So it will be like, as such, 335 base pair. Now come to the other assay for 5B. Similarly, wild type will be digested into 304 and 32 base pair. We won't be able to see this segment because it's smaller again. And then 5B won't be digested because there's a mutation. Now, because the smaller chunk will appear, this will move faster compared to the bigger chunk. So you will expect to see 5b somewhere here compared to the wild type. Similarly for the cry2 Now, let's see the pattern. How did we load the sample on the well? So we follow the same way as it's written here: A1, B1 and W1 for assay 1, and then A2, B2, W2 for assay 2. So this is the gel lock machine. I have put the gel inside it along with the ladder, the scale, so that we can see how millimeter our DNA has migrated. Now this has the UV lamp, and because we have put a dye which intercalates with the DNA, and in the presence of UV light you can visualize that DNA. So let's on the UV light. So these are the modes through which you can adjust the brightness, and you can increase the size of the, you can zoom in basically, but I don't want to interrupt the settings. I think it is clear now. So at the bottom there are the wells of the gel where we loaded our DNA, and this is the DNA standard, the known DNA sizes, and this particular size is 300 base pair, and all our samples are near the 300. So this chunk is for assay 1, which is cry assay, and this is A1, B1 and wild type. Similarly, this is for the second assay, A2, B2 and wild type. So if you look closely, you'll see these two bands are quite smaller compared to the middle band here, because this is not digested, and we assume that this one is cry mutant. Now if you come here, which one is smaller here? These two look smaller compared to this one, and this one actually 1 base pair bigger than the cry2. The size of this one is 336 and here is 335. Yes, so this we assume that this represents phytochrome B mutant. And you see a ladder here, so you can see how much our DNA is migrated in a particular given time. So yes. So now we are going to see morphology of these mutants along with the wild type. Let's start with the wild type. So as you can see, it started flowering. Flowers are here, beautiful, and it's growing well. See this genotype. Compared to the wild type, the stem is elongated, and this is also flowering. But I think if you see the greenery, I'm not sure if it is clear from there, but this is light green. Come to the other genotype. Oh, this is very small actually, and it delayed in flowering. None of the plants are flowering now. And leaf-wise, it looks more dense, packed. So I think there are more leaves compared to these two. So let me take one plant from each genotype and we'll describe their rosette leaves. So this is the wild type. Yeah. So this area is called rosette area, and the small spoon-shaped leaves are the colline leaves. So I'll remove one by one and we'll see how many rosette leaves are there. So, okay, there's also cotyledons. They are like immature leaves, first emerging leaves, embryonic leaves. So we won't count that. Let's start with the true leaves. So these leaves, they don't have petiole, and we won't consider that as a rosette leaf. So these are the leaves from this wild type genotype. Let's go to the other genotype. This is 1A, sorry. So I'll take this plant out. Yeah. So it looks like it has less leaves, less counted. So 1, 2, 3, 4, 5, and 6. That's it. Now time for the other genotype. Okay, insect has eaten this one, so I have to take another one. This is healthy plant. So it looks like it has more leaves, as counted. Two, 3, 4, 7, 8, 9, and I'm not sure these ones are from the rosette leaf or they are the choline, because they are about to flower, so maybe by the time they will convert it into the rosette leaf, we are not sure, so we won't count this. So these are the 3 genotypes, and based on the rosette leaf count, I think B has the more leaves, and this one is also delayed in flowering, and A has less leaves, rosette leaves, and it's early in flowering, and the stem side is also elongated compared to the wild type. So now I'm going to take picture of these plants which I dissected. So the first thing is align them and give proper names, because later on you won't know which is which genotype. So proper naming is required, and when you're sure with their alignment, you can take picture. Sorry, I'm using my pocket phone. It's good to use professional's camera, I guess, but you can also take picture using your camera when you use right settings. So because I'm using black background, so it will automatically adjust the brightness. So you need to adjust the brightness of your phone. You don't need too dark or too light picture. So I'm just adjusting the image, light, and then when you're sure everything is focused and everything is inside the frame, click it. And it's done. I'm not sure it's visible.